the cells holding today's memories are still working. While you lie here, your hippocampus is quietly replaying fragments of what happened to you today. It is a small curved region buried deep in the middle of your brain, and it never stops. A conversation, a face, the route you took home. Each fragment passes through its circuits again, strengthening the connections that will survive into tomorrow and allowing others to weaken. This process runs every night without asking your permission, and it runs so efficiently that you will wake with no memory of it having occurred. The work is invisible, the work is constant, and the neurons doing it are the most active and energy-hungry cells in your brain. They do not rest, not even during sleep. They are also, under certain conditions, among the most vulnerable cells you carry. But here is what is strange. In many people, something else is also happening inside those same neurons, and it began years or even decades before any symptom appeared. It produced no signal, nothing that could be felt, no headache, no change in energy, no warning of any kind. The brain compensates so effectively that it can lose a substantial fraction of its cells in certain regions before the first name slips, before the first appointment is forgotten, before anyone around them notices anything is wrong. The damage is invisible too for a very long time, but it is not random and it is not silent by chance. The biology that makes compensation possible is the same biology that is slowly being exhausted by the process it is compensating for. And when the damage does finally declare itself, it moves through the brain in a sequence so specific that researchers who have studied enough post-mortem tissue can map its path. The same region goes first, the same region goes second, in person after person, across different lives and different decades the pattern holds. This regularity is not coincidence, it is a signature of a cause. Alzheimer's disease is not a random failure of aging tissue. The neurons it takes first are not the old or the tired ones. They are the cells encoding today's experiences, the most active and energy-hungry neurons in the network. 1 in 10 people over the age of 65 will live this. Most of them have no idea it has already begun. And Alzheimer's is not alone. Parkinson's disease follows the same principle, so does ALS, and so does Huntington's disease, along with dozens of related conditions. In each of them, specific populations of neurons begin to fail in a specific order. In each of them, the order is not random. The most active cells carry the highest load. The highest load comes due first. How does a protein that has done its job correctly for decades begin recruiting others into the same mistake? And why, when the damage finally declares itself, does it always follow the same route through the brain? If you enjoy exploring questions like these about the machinery behind the mind, a like would be highly appreciated. And if tonight's episode raises a thought or a question you want to share, the comments are there for exactly that. With that said, let's return to those questions. Both questions start from the same place. What a neuron is built to do, and what sustaining that across a lifetime leaves behind. To understand why neurons fail in neurodegeneration, you have to start with what neurons are asked to do. The first thing that makes neurons unusual is that most of them are yours for life. Almost every other cell type in your body is replaced on a schedule. Skin cells last weeks, red blood cells last a few months. The cells lining your gut are replaced in days. But the neurons in your hippocampus and cortex, along with those in the deep brainstem structures most implicated in neurodegeneration, were formed before you were born. They have been running without replacement ever since. There is some limited growth of new neurons in a few specific regions, but it is modest. For the cells most vulnerable to these diseases, there is no fresh supply waiting in reserve. Every year of use adds to the total damage they must manage. Every year of managing it adds to the conditions that make management harder. The second thing is what that running demands. Your brain is about 2% of your body's weight, but it consumes close to 1/5 of all the energy your body produces. Every neuron runs on a constant supply of glucose and oxygen, and if that supply is interrupted for even a few minutes, cells begin to die. Most of this energy goes into something specific, maintaining the electrical balance across the cell membrane that makes firing possible. A neuron is not resting between signals. It is actively working to preserve the conditions that make the next signal possible, and that work never stops, even during sleep. The price of this constant activity is a constant production of chemical waste. These byproducts of energy generation are damaging to the molecules around them, including proteins. The cell has systems to manage them, but these systems are imperfect. Over decades, some of the damage they cause accumulates in cells that cannot divide to dilute it. The third thing is the axon. An axon is the projection a neuron uses to communicate with distant cells. The motor neurons running from your spinal cord to your legs can extend for more than a meter. The nucleus of that neuron sits at one end, and it must manufacture proteins and ship them down that entire length to maintain the axon's function. This internal delivery system, called axonal transport, operates continuously. Thousands of different proteins move in both directions simultaneously, carried along tiny tracks called microtubules. The system depends on those tracks staying intact. Anything that disrupts the tracks or the motor proteins that carry cargo along them slows delivery to the far end of the cell and eventually starves it. A motor neuron extending to your foot is asking its nucleus to keep a distant outpost supplied across a distance that, at the scale of cells, is equivalent to running a supply line across a continent. If the delivery system slows, the far end begins to fail before anything near the nucleus shows signs of trouble. And this is where all three vulnerabilities converge. Neurons synthesize enormous quantities of protein every day to replace worn components, to build and rebuild synapses, to maintain the tracks of the internal transport system. Every protein produced carries a risk, the risk of emerging in the wrong shape. A cell with a short lifespan can tolerate a higher error rate because its descendants start fresh. A neuron that may live for 80 or 90 years cannot. Every misfolded protein it fails to catch is still present decades later, a small addition to an accumulating load. The question is not whether errors occur. They occur in every cell that ever lived. The question is whether the systems catching and correcting them can keep pace. In neurons, the consequences of falling behind are higher than in almost any other tissue in the body, and they persist longer. There is one more thing worth knowing. Neurons are exceptionally sensitive to the shape of the proteins they depend on. The proteins that build synapses, keep the transport tracks open, and regulate how signals pass between cells each need to be exactly right. A protein that arrives in the wrong shape does not simply fail to help. It can actively block a process that would otherwise have worked. In a cell that will run for 80 years without a replacement arriving, errors that are not caught do not disappear. They stay. Every second of your waking life, the neurons in your brain are producing proteins at a rate that would be staggering to watch if you could. Proteins are not simple molecules. They begin as long strands of amino acids, assembled one by one according to the instructions encoded in your DNA. But the strand is not the finished product. To become functional, it must fold. It must collapse from a long, floppy shape into a precise three-dimensional structure, a structure determined by the sequence of amino acids and by the forces they exert on one another as the strand bends and twists and searches for its lowest energy state. The finished shape is not approximate. For most proteins, only one configuration out of an enormous number of possible configurations allows the protein to do its job. The wrong shape means the protein cannot bind to what it needs to bind to, cannot carry out the reactions it needs to carry out, cannot perform its function. Think of a combination lock, the kind with a series of numbered dials, each of which must click to the correct digit before the mechanism releases. The lock does not open at almost correct. Every dial must reach its position for anything to happen. A protein fold works something like this. The molecule passes through a sequence of intermediate shapes, each one constraining what can happen next, until it arrives at the one configuration where all the internal forces balance simultaneously. The path matters as much as the destination. A strand that takes a wrong turn early will carry that error through every subsequent step. The cell does not leave this process to chance. It employs a class of proteins called molecular chaperones, whose entire purpose is to monitor newly forming protein strands and guide them toward the correct fold. Chaperones recognize when a strand has gone wrong, seize it before it can interact harmfully with other molecules, and either shepherd it toward the correct shape or flag it for destruction. The cell also maintains a molecular shredder called the proteasome, which breaks misfolded proteins into their constituent parts, releasing the amino acids to be used again. Under normal conditions, these systems are extremely effective. Most proteins that emerge wrong are caught and dealt with before they cause harm. But the system has a key vulnerability. A protein strand that has not yet finished folding exposes sticky inner regions that in the finished molecule would be buried safely out of reach. In a correctly folded protein, these regions are locked inside. In a misfolded one, they are exposed, and exposed sticky regions will grab onto the nearest available surface. This is not a flaw, it is physics. Two unfinished molecules with exposed sticky regions will cling to each other. They will pull others in. The resulting cluster is sticky, disorganized, and increasingly resistant to the chaperones trying to dismantle it. This is the first step toward aggregation, and it is happening in every cell in your body all the time at low rates that the quality control systems have been managing without incident for most of your life. A healthy young cell deals with this constantly. Every minute some proteins emerge wrong. Most are caught within seconds and either fixed or destroyed before they can cause damage. The system is fast, efficient, and largely invisible in its success. You never notice the thousands of errors being silently corrected. You only ever see the ones that get through. The critical variable is not whether misfolding occurs, it is whether the rate of misfolding stays within what the cleanup systems can handle. When that balance begins to tip, the consequences are not immediate, they are cumulative, and they are specific. Different proteins misfold in different ways, aggregate into different structures, and target different cell types. The type of aggregation determines the disease. The location of the affected cells determines the symptoms. But the underlying logic is the same. Something that should fold correctly did not, and the system that should have caught it could not keep up. The helper protein network is more extensive than it might seem. There are dozens of different chaperone proteins, each specialized for different stages of the folding process. Some catch newly made strands before they have even finished being assembled. The system has been solving this problem across billions of years of evolution. What changes with age is not its design, it is its capacity. A neuron that fires constantly produces far more protein than one that is mostly quiet. More production means more chances for misfolding, and more misfolded proteins arriving faster than the chaperone network was built to handle at once. This is why the most active neurons carry the highest load. Not because they are weaker, because they are busier. So what happens when a misfolded protein escapes the quality control systems? For a time, nothing visible. The cell produces misfolded proteins constantly and deals with most of them before they accumulate, but occasionally one slips through. It exposes its sticky interior to the cellular environment, and when it does not just float harmlessly, it grabs. Think of a snowball sitting in an open snowfield, not rolling, sitting. Snowflakes settle on its surface and cling. More settle on those. The larger the ball becomes, the more surface there is to catch the next one. Eventually, the mass is large enough to trap flakes that would have drifted past something smaller. It does not move. The snow comes to it. Misfolded proteins aggregate in a similar way. A single misfolded molecule with its sticky inner surfaces now exposed attracts other molecules to its surface. Some of those molecules are also misfolded. Others are normal proteins that happen to touch the exposed surface and find themselves stuck. The mass grows. At first, it forms small clusters, just a handful of molecules joined together. These tiny clusters are the most damaging stage of aggregation, for reasons that will become clear. Over time, the small clusters grow into longer threads. The threads then assemble into larger, more stable deposits. This pattern is not a peculiarity of one disease. It is a common feature of many neurodegenerative conditions. What differs between them is which protein is involved and where in the brain it accumulates. In Alzheimer's disease, two different proteins aggregate in two different places, and together they create a condition that systematically dismantles the part of the brain most responsible for being you. To understand why, you need to understand each one separately. But before going further, there is something worth pausing on. The sticky surface of a misfolded protein is not chemically inert. The oligomers it forms are not merely obstructions, they are reactive. They embed themselves in cell membranes. They generate oxidative stress. They interfere with the normal signaling proteins need to carry out at synapses. In many neurodegenerative diseases, the most acute damage is not done by the large visible deposits. It is done by the small, soluble oligomers that form long before anything becomes visible under a microscope. By the time the aggregation is detectable from the outside, the disruption of cellular function has been building for years. This is one reason why the diseases are so difficult to catch early, and why the window between onset and diagnosis has historically been measured in decades rather than months. And the timeline deserves attention. Researchers studying blood and spinal fluid have found evidence of amyloid-beta accumulation in people who are thinking clearly, who have no symptoms, who will not receive a diagnosis for another 10 or 15 years. The aggregation is not the disease, it is the prelude to the disease. Neurons begin to compensate. They prune connections, recruit backup circuits, and push their protein quality control systems harder. This compensation is effective for a while. It is also exhausting. The cells working hardest to maintain function are the cells accumulating the most damage in the process. And when their reserves are finally spent, the decline that follows can appear sudden to everyone watching, even though the damage behind it had been building for years. This long silent period has reshaped how researchers think about when intervention would need to happen to be effective. Waiting until symptoms appear means waiting until the brain has already lost a significant fraction of the neurons most central to its function. The visible decline is not the beginning of the disease. It is the point where compensation has finally exhausted itself. Some researchers now speak of treating neurodegeneration in the same terms as treating cardiovascular disease. The intervention that matters is the one that begins before any event occurs, while the balance can still be shifted rather than merely slowed. The biology of the silent decade is, in this sense, not a tragedy. It is a window. The question is whether it can be kept open long enough to climb through. Alzheimer's disease involves two different proteins failing in two different places. The first is a small protein fragment called amyloid-beta, and to understand what it does you need to understand what it is. Amyloid-beta is not a foreign invader. It is produced by your own neurons as part of normal cellular metabolism. Every day your brain cells process a larger protein, cutting it into fragments using two molecular scissors. Most of the time this cutting produces fragments that are easily cleared, but one particular cutting pattern produces amyloid-beta fragments that are stickier than the others. These are not swept away as efficiently. They drift out of neurons into the fluid-filled spaces between cells, where they have a tendency to find each other and aggregate. Alois Alzheimer noticed them in 1906 when he examined the brain of a patient named Auguste Dieter under a microscope. She had died in her mid-50s after years of memory loss, confusion, and language difficulties that had puzzled her physicians. What Alzheimer saw in her brain tissue were dense deposits between the neurons, deposits that today we call senile plaques, and twisted threads inside the neurons themselves. He understood that he was looking at something no one had properly described before. What he could not have known was that the deposits themselves might not be the primary weapon. For decades after his discovery, the assumption was that the plaques themselves were the main driver of neuronal death. These large deposits of amyloid-beta seemed the obvious culprit. Treatments were designed to clear them. Clinical trials were run to test drugs that successfully reduced plaque burden in patients' brains. Many of those trials failed to produce meaningful cognitive benefit. The plaques were going. The decline was not slowing. The resolution came from a closer look at what amyloid-beta does before it forms visible plaques. The small soluble oligomers, clusters of just a handful of amyloid-beta molecules, turned out to be far more biologically active than the large aggregates they would eventually become. Think of sand inside a watch mechanism, invisible until something goes wrong, too fine to be easily removed, but enough to grind the gears apart over time. The oligomers insert themselves into cell membranes. They interfere directly with the receptors at synapses that neurons use to communicate. They trigger oxidative stress and disrupt the mitochondria that power the cell. The synapse is where this damage becomes a disease you can observe. A synapse is the gap between two neurons across which chemical signals pass, and it is the physical substrate of everything you have learned and remembered. A neuron that is having its synapses disrupted by amyloid oligomers cannot communicate efficiently with its neighbours. The connections that should strengthen through use weaken instead. The network begins to lose precision, and this process, happening simultaneously at countless synapses across the hippocampus and prefrontal cortex, is what you experience as the progressive difficulty with new memories that is usually the first symptom of Alzheimer's disease. The large plaques accumulating nearby are a marker of how long the process has been running, not the agent doing the acute damage. The reason this matters beyond the molecular level is that a synapse is not an abstract unit. It is the physical location where one thought connects to another, where a name links to a face, where the path from a hallway to a room links to everything that happened in it. When the synapses in the hippocampus begin failing, the experience is not of blanket forgetting. It is of connections becoming unreliable. A name that was retrievable yesterday may not be retrievable today. A face that felt familiar becomes unfamiliar. Recent events fade faster than old ones, because recent memories are encoded in the synapses that are most actively maintained, and those are the ones most exposed to the accumulating oligomers. The oldest memories, encoded in more distributed networks across the cortex, hold longer. They are upstream of the damage. The person often retains their childhood while losing yesterday, which is one of the details that makes the disease so difficult to witness from the outside. And the neurons hit first are not random ones. They are the neurons of the hippocampus. These are among the most active cells in the brain. They produce the most amyloid-beta and are exposed to the highest concentration of oligomers. The damage begins where the work is hardest. While amyloid-beta does its damage outside neurons, in the spaces between them, a second protein is failing simultaneously from within. Neurons, like all cells, maintain an internal transport network. Imagine a canal system that runs through the cell, with long, navigable channels extending down the full length of the axon, all the way from the nucleus in the cell body to the terminals at the far end. Along these channels, molecular motors carry cargo in both directions. The cargo includes proteins and organelles, neurotransmitters, and the molecular equipment needed to keep the distant parts of the cell alive. The channels are built from structures called microtubules, and those structures require constant maintenance to stay functional. The protein responsible for maintaining them is called tau. Tau works by binding to microtubules at regular intervals like ties on a railway, keeping the tracks spaced correctly and stable enough to carry traffic. A neuron with healthy tau maintains open channels, cargo flows, the far end of the axon receives what it needs, the cell stays alive. In Alzheimer's disease, tau undergoes a chemical change that causes it to lose its grip on the tracks. It picks up far more chemical tags than it should carry, and that extra load causes it to let go. Once detached, tau does not simply dissolve into the cellular fluid. It behaves like every other misfolded and displaced protein. It finds other tau molecules, sticks to them, and begins to aggregate. The resulting tangles form inside the neuron, filling the cell body and crowding into the axon, taking up space and further disrupting whatever transport infrastructure has survived. The effect on the neuron is twofold. The microtubule tracks, no longer maintained, begin to fall apart. The canal walls collapse. Cargo that should be reaching the far end of the axon does not arrive. The synaptic terminals, already stressed by amyloid oligomers on the outside, are now also being starved from within. The neuron is failing on two fronts simultaneously, and the combined pressure is more than the cell's reserves can sustain indefinitely. A German neurologist named Alois Alzheimer saw both problems in Augusta Dieter's tissue in 1906. The plaques between the neurons and the twisted threads inside them. He was looking at the end stage of a process that had been running for years. The threads, we now know, follow a trajectory of their own. They appear first in a small region called the entorhinal cortex, one of the earlier stations in the brain's memory circuit. They spread next into the hippocampus, then into the association cortex, and finally into the broad neocortex. This progression was mapped with great patience by a German neuroanatomist named Heiko Braak, who examined the brains of hundreds of people who had died at different points in the disease's course. His maps showed a staging system that was nearly universal, the same regions in the same order, across cases separated by decades and continents. He had found the disease's root. What he had not yet explained was why it followed one. But the fact that a root existed at all was itself significant. It meant that disease was not arriving randomly at different regions of the brain. It was moving through them in sequence like a fire spreading along a fuse, and the fuse was the brain's own wiring. The significance of Braak's staging was not only medical, it changed what people thought they were looking for. Before his maps, the assumption was that Alzheimer's disease was primarily a chemistry problem. Find the chemical that accumulates, develop a drug to remove it, and the progression should stop. His maps revealed that the disease was also a network problem. The pattern of spread was not determined by the chemistry of amyloid or tau alone. It was determined by the anatomy of the brain, by which regions communicate most densely with which other regions, by which neurons have the most synaptic contacts with already affected cells. Treating the chemistry without addressing the network dynamics would be like removing the water from one section of a flooded canal while leaving all the channels open upstream. The source and the root both matter. Understanding the root was the necessary first step to understanding why the source is so hard to stop. Heiko Braak's maps raised a question that took years to properly answer. If tau tangles begin in the entorhinal cortex and move outward from there, what is moving them? The entorhinal cortex does not simply become diseased and then sit still while adjacent regions spontaneously develop the same disease. The spread follows anatomical connections. The regions that receive tau pathology after the entorhinal cortex are precisely the regions that the entorhinal cortex connects to most directly. The disease appears to travel along synaptic routes, passing from one neuron to the next through the very channels neurons use to communicate. To understand how, you need to encounter one of the most counterintuitive discoveries in modern neuroscience: that a misfolded protein can instruct a normal protein to become misfolded in its image. Think of someone teaching origami from a folded model that has a crease in the wrong place. Everyone who learns from that model copies the wrong crease. Now they each teach someone else. The wrong fold spreads through the room, not because anyone intends it, but because the wrong shape is now the template. Each new person who learns from it becomes another source of the mistake. Misfolded proteins behave as seeds in precisely this way. A molecule of tau in the wrong conformation does not simply stick to other tau molecules at random. It acts as a template. A normal tau molecule that comes close enough to the misfolded one is recruited into the same conformation, coaxed from its correct shape into a copy of the wrong one. That newly misfolded molecule is now also a template. It recruits others. The process is self-amplifying. This mechanism was once thought to be unique to a class of diseases caused by a protein called a prion, the same mechanism responsible for mad cow disease and for a rare human condition called Creutzfeldt-Jakob disease. What has become clear over the past 2 decades is that the prion principle is not a peculiarity. It is a general feature of protein aggregation in neurodegeneration. Tau in Alzheimer's disease spreads through the brain like this. Alpha-synuclein in Parkinson's disease spreads like this. TDP-43 in ALS spreads like this. The protein differs, the regions affected differ, the symptoms differ. The mechanism is the same. Think about what this means for Braak's maps. The staging is not a coincidence of geography. 2 neighbouring regions happening to develop similar disease independently. It is transmission. The entorhinal cortex seeds the hippocampus because it sends axons into the hippocampus. The hippocampus seeds the association cortex because it connects there. The pathology follows the wiring of the brain, region by region, along routes that took decades to build and that now carry something they were not built to carry. This is why the disease has a predictable order. The structure of the brain determines the route. The route determines the sequence of loss. The discovery that this mechanism was real came largely through the work of a neurologist and biochemist named Stanley Prusiner. He spent years in the 1970s and 80s arguing that the infectious agent behind a family of devastating brain diseases was not a virus or a bacterium, it was a protein. The scientific community resisted the idea. A protein that reproduced by converting other proteins into copies of itself, without any genetic material of its own, seemed to break the rules of how disease worked. Prusiner called the agent a prion, and he was awarded the Nobel Prize in Physiology or Medicine in 1997 for the discovery. What the subsequent decades made clear was that this mechanism is not a rare exception. It is a general principle of how these proteins behave. Every major neurodegenerative disease involves a protein that spreads this way. The specific protein differs. The brain region where it starts differs. The symptoms differ. But the underlying process is the same. One wrongly folded molecule becomes the template for the next, and the next becomes the template for the one after that. The disease does not stay where it starts. The aggregation of misfolded proteins is not just chemistry going wrong inside one cell. It is a process that travels, one that follows the connections the brain has spent a lifetime building. By the time the protein has spread from one region to the next, the problem is no longer contained. Addressing only the cells where it started will not stop it from continuing where it has already arrived. Your brain is not defenseless against protein aggregation. It has an immune system, and that immune system is watching. About 10 to 15% of all brain cells are microglia, a class of immune cell that lives permanently in the brain tissue and patrols it continuously. Microglia are mobile. They extend and retract long, thin projections through the spaces between neurons, sampling the local environment for signs of damage. Under normal conditions, they clear cellular debris and trim synaptic connections that are no longer being used. They remove molecular waste before it can accumulate. They are the brain's maintenance crew. When amyloid plaques begin to form, microglia respond. They cluster around the deposits and attempt to engulf and break them down. This response is not negligible. Without functional microglia, amyloid accumulation would be faster and more widespread. People who carry a gene variant that weakens this microglial cleanup ability face a significantly higher risk of Alzheimer's disease. But the plaques are large, chemically stable, and resistant to being cleared in this way. Microglia that attempt to engulf them and fail do not simply give up. They activate. They release chemical alarm signals that call other immune cells to the site and increase the intensity of the local response. In a wound or an infection, this escalation is appropriate. It recruits reinforcements, accelerates clearing, and then subsides when the threat is resolved. In neurodegeneration, the threat is not resolved. The amyloid keeps accumulating. The tau tangles keep forming. The microglia remain activated, and the inflammatory signals they produce keep circulating through the brain tissue. Imagine a scratch that never fully heals. Every time the skin begins to close, it is disturbed again, so the tissue around it never leaves the inflamed state that supports repair. The inflammation that should be temporary becomes permanent, and inflammatory tissue is not neutral. The same alarm signals that were meant to coordinate a response also damage the very neurons they surround. At sufficient concentrations, for sufficient duration, they are toxic. This chronic low-grade inflammation, sometimes called neuroinflammation, accelerates the death of neurons that are already under stress from amyloid oligomers and tau tangles. It also disrupts the function of neighbouring neurons that have not yet accumulated significant aggregates. The inflammatory environment lowers the threshold at which otherwise manageable protein stress becomes lethal. A neuron that might have survived another decade is instead lost within years. Here is what makes this particularly difficult to intervene in. The microglial response is both protective and destructive, and the same cells are responsible for both. You cannot simply switch them off. Suppress the response entirely, and amyloid clearance fails faster, the deposits grow, and the disease progresses without the one system that was slowing it. Allow it to continue unchecked, and the inflammation that was meant to protect the brain ends up accelerating the damage instead. The question of how to adjust this response without removing it is one of the active areas of research in the field, and it remains genuinely unresolved. What makes neuroinflammation particularly relevant to the question of timing is that it appears to accelerate not just at the site of existing pathology, but throughout the brain. The alarm signals released by activated microglia travel through the fluid that flows through the brain and can affect neurons far from where they were produced. A neuron in a region not yet reached by amyloid or tau may already be functioning in an inflammatory environment that lowers its tolerance for additional stress. When the protein aggregation eventually arrives, it arrives in tissue that has been primed. The threshold between manageable and lethal is already lower than it should be. The cell has two main systems for dealing with damaged and misfolded proteins, and both of them matter enormously in neurodegeneration. The first is the proteasome. This is a molecular machine that sits inside the cell and functions as a shredder. Proteins flagged for destruction are tagged with a small marker molecule that sends them to the shredder. There they are broken down into their basic parts, which can then be recycled. The system operates continuously. In a healthy neuron, most misfolded proteins are caught by chaperones and sent to the proteasome within minutes of being produced. The shredder runs fast enough to keep pace with the rate of error. Think of a city office that generates confidential documents all day long. Each document goes into an in-tray. At the end of every hour, the in-tray is emptied into the shredder. For most of the day, the shredder keeps up with the output. But if the shredder's motor begins to slow, the documents accumulate. The in-tray fills. Documents that should have been destroyed 10 hours ago are still sitting in the pile. Some of them find their way out into the office, where they create problems they were never supposed to. The second system is autophagy. Autophagy is a more general process in which the cell forms a membrane sac around a target and digests its contents. It handles things the proteasome cannot. Larger protein aggregates, entire organelles that have become damaged or dysfunctional, including mitochondria. When a power plant in a neuron starts generating more damage than energy, autophagy identifies it and removes it before it causes further harm. This removal of damaged power plants is critical for cells that cannot afford to keep running on broken equipment. Both systems decline with age. The shredder becomes less active. The autophagy machinery is produced more slowly and regulated less precisely. And in some neurodegenerative diseases, the aggregating proteins directly impair the very systems that should clear them. This is where the situation turns particularly difficult. The aggregates form, they damage the cleanup systems. The damaged cleanup systems allow more aggregates to form. The load grows faster than the weakened system can handle. In a cell where this cycle runs for years, the accumulation eventually becomes impossible to manage. A system that was already running at reduced capacity is being sabotaged by the very problem it is trying to solve. Think of a cleaning crew whose tools keep breaking down because of the very mess they are trying to clear. Every hour they spend working to fix their equipment is an hour the backlog is growing, and the material damaging their tools is the same material they were brought in to remove. A system under this kind of pressure does not decline gradually toward a fixed level and hold there. It declines toward a level that makes further decline more likely. The speed of accumulation increases as the capacity to manage it falls. Yoshinori Osumi, who received the Nobel Prize in Physiology or Medicine in 2016 for his work on autophagy, spent years at the National Institute for Basic Biology in Japan studying what happened when the cellular digestion system was disabled in yeast. He found that even cells under starvation, cells that desperately needed to recycle their own components, could not do so without autophagy. What his work eventually helped establish was that maintaining autophagy in aging neurons is not a luxury. It is the difference between a cell that can manage its accumulated damage and one that cannot. There is a detail about Parkinson's disease that makes the connection between autophagy and neurodegeneration very clear. In some families where Parkinson's runs through generations, the genetic mutations responsible turn out to affect exactly this system: the tagging and removal of damaged power plants. When that process breaks down, damaged mitochondria accumulate in dopamine neurons. Mitochondria that should have been cleared keep generating chemical waste. The neurons they are supposed to be powering are instead being worn down from inside. The genetics pointed directly at the pathway, and the mechanism behind those inherited cases turned out to be the same one running in people with no family history at all, simply arriving later when the age-related decline in autophagy finally pushed the balance past the same point. Alzheimer's disease is the most common form of neurodegeneration, but it is not the only one, and understanding the others reveals how deep the shared pattern runs. Parkinson's disease affects about 1% of people over the age of 60. Its most recognizable early symptom is a tremor at rest, a rhythmic shaking of the hands or limbs that occurs when the muscles are not being used and subsides when movement begins. Beneath this symptom lies a specific catastrophe. The neurons in a region called the substantia nigra, a small cluster of cells in the midbrain that produce a neurotransmitter called dopamine, are dying. The loss of these cells reduces the dopamine available to the circuits that coordinate smooth movement. The result is the characteristic rigidity and slowness of the condition, along with the tremor. The cause follows the same principle as Alzheimer's, a protein called alpha-synuclein, normally found at synaptic terminals where it plays a role in neurotransmitter release, misfolds and aggregates. The aggregates form large deposits inside neurons called Lewy bodies. Braak and his colleagues produced a staging map for alpha-synuclein pathology in Parkinson's disease, just as he had done for tau in Alzheimer's, and found the same pattern. The aggregates appear first in the brainstem and the smell processing region. From there they spread upward into the midbrain and eventually the cortex. The spread follows synaptic connections. It moves like tau moves, recruiting normal protein into the wrong shape as it goes. ALS, or amyotrophic lateral sclerosis, destroys motor neurons, the cells that carry movement signals from the brain and spinal cord to the muscles. The proteins most commonly involved help manage the cell's genetic instructions. When they misfold and aggregate inside motor neurons, the cells lose the ability to process those instructions and begin to die. The muscles they served become paralyzed. In most forms of ALS, the disease spreads from its site of onset through the motor neuron network, again following the wiring. ALS typically begins in one region and spreads outward from there. A person might notice one hand becoming weaker before the other, or find that the muscles controlling speech begin to fail, while movement in the legs is still intact. The progression follows the motor neuron network, spreading from the cells nearest the starting point through the connections they share with adjacent neurons. The disease does not jump at random. It follows the wiring. Huntington's disease is genetically determined rather than sporadic, caused by an expansion in a single gene that produces a version of the Huntington protein with an abnormally long region. This extended region misfolds. The aggregates that form preferentially affect neurons in the striatum, a structure involved in movement planning and habit formation. The disease causes involuntary movements and cognitive changes, along with psychiatric symptoms, and it begins earlier in life than most other neurodegenerative conditions. What connects them all is not the protein, not the brain region, not the symptom. What connects them is the mechanism. A protein that should fold correctly does not. It forms aggregates that spread from cell to cell and overwhelm the cleanup systems that should manage it. Neurons that cannot be replaced begin to die in a sequence that follows the brain's own wiring. There is a frontier condition that illustrates the commonality even more clearly. Frontotemporal dementia affects the frontal and temporal lobes, the regions most involved in personality and language and social judgment. It causes changes in behavior and personality that often arrive before any obvious memory loss, which is why it is sometimes diagnosed as a psychiatric condition before the neurological cause is found. Some forms of FTD involve tau, others involve the same protein implicated in ALS. A significant number of people have both ALS and FTD simultaneously, a combination that reveals something important. What determines the disease's symptoms is not which protein is involved, but which neurons it reaches first. The same misfolded protein that destroys motor neurons in ALS destroys frontal neurons in FTD, depending entirely on where the aggregation began. The protein is the same. The starting point makes the disease. This is both a complication and a clarification. A complication because it means there is no simple one-to-one mapping between protein and disease. A clarification because it confirms that the underlying mechanism is genuinely shared, and that understanding it in one context carries knowledge directly into all the others. If protein aggregation is the mechanism, and failing cleanup systems are the failure mode, the remaining question is why this happens in aging brains and not in young ones. The issue is not that young neurons are more careful and old ones become careless. What changes with age is not the accuracy of protein production. What changes is the capacity of the response to errors. In a young cell, the chaperone proteins are numerous and active. The proteasome runs at close to full speed. Autophagy is initiated efficiently and completed reliably. The response is fast enough that misfolded proteins are cleared before they have time to find each other and aggregate. the balance holds. With age, every component of this system gradually tips. Think of a boat crossing a wide estuary. There is a slow leak in the hull, there has always been a slow leak, and the bilge pump has always cleared it comfortably. The boat crosses without difficulty. But over many years, the leak widens slightly and the pump motor wears down. Neither change is catastrophic on its own, but at some point the rate of inflow exceeds the rate of drainage. The boat does not sink immediately. The water rises slowly, and for a long time the consequences are invisible to anyone watching from the shore. Then the water reaches the engine compartment. Several things contribute to this tipping. The power plants inside aging neurons produce more chemical waste and generate less energy than they once did. That chemical waste directly damages proteins, creating more misfolded molecules than a young cell would produce. Errors in copying genetic instructions accumulate over decades, affecting how accurately proteins are built in the first place. At the same time, the cleanup side weakens. The shredder itself is damaged by that same chemical waste and becomes less efficient at doing its job. The autophagy system becomes less responsive with age, clearing things more slowly and less completely. Each of these changes is modest on its own. None of them stands out on its own, but they all move in the same direction at the same time, and collectively they shift the balance between the rate at which problems arise and the rate at which they can be cleared. At some point that balance runs to the edge of what the cell can manage, and then there is genetics. For most people, the timing of this tipping is not fixed. It is modulated by variants in genes that affect how efficiently amyloid-beta is cleared from the brain, how active the microglial response is, how robust the autophagy pathway remains with age. The most significant known genetic risk factor for late-onset Alzheimer's disease is a variant of a gene called APOE. One version of this gene makes it harder for the brain to clear amyloid-beta from the fluid between cells. People who carry 2 copies of it face a substantially elevated lifetime risk. People who carry no copies face a lower than average risk. The gene does not cause the disease. It shifts when the tipping point arrives, earlier or later. For people with rare inherited mutations that cause amyloid-beta to be overproduced from early in life, the tipping happens regardless of age. These familial forms of Alzheimer's can begin in the 40s or 50s, decades before the more common form typically appears. They reveal what happens when the production side of the equation is shifted rather than the clearance side. The endpoint is the same. The protein accumulates faster than it can be cleared. The spread begins. The neurons follow the route that Braak mapped. Familial cases do not represent a different disease. They are the same disease running on an accelerated timeline. What the genetics collectively show is that the tipping point is not fixed. It is determined by the balance between how fast problematic proteins accumulate and how efficiently the clearing systems can respond. Shift either side and you shift the timing. Inherited mutations can push the accumulation side up, flooding the brain with amyloid-beta earlier than the clearing systems can handle. Certain gene variants pull the clearance side down, making the brain less efficient at removing what builds up. And the slow age-related decline in the shredder and autophagy does the same thing, gradually and in everyone. This means that neurodegeneration is not a disease that arrives from outside. It is the outcome of a balance failing, a balance that every brain is maintaining right now. Most brains will maintain it successfully for a lifetime. Some will not, and the reason is not a mystery imposed from outside. It is the accumulated consequence of specific biological variables, most of which are now at least partially understood. And that understanding, still incomplete as it is more than enough to make the biology feel personal. Because it is personal. The balance is yours. Come back for a moment to where this episode started. Your hippocampus is working. Right now, while you rest, the neurons in that curved structure are replaying the day's events. They are strengthening the connections that encode what you will remember and allowing others to fade. The same neurons that opened this episode are still active, and now you know something about what that activity involves. Every one of those neurons is continuously synthesizing proteins. Every protein that emerges must fold correctly. Most do. A small fraction do not, and those are being caught by chaperone proteins and either refolded or sent to the proteasome. Autophagy continuously runs through the cell body. They clear aggregates that have built up, remove mitochondria that have become inefficient, and maintain the internal transport tracks that keep the far ends of the axons alive. Microglia are moving through the surrounding tissue, sampling for debris and removing fragments that should not be there. They manage the inflammatory state of the surrounding tissue. The whole system is running right now as you listen to this. The scale of this is hard to hold in mind. In a single neuron in your hippocampus, thousands of protein synthesis events are occurring every minute. Each one carries the same small risk of producing a misfolded protein. The systems running to intercept those errors are not a recent invention. The chaperone network and the autophagy machinery predate the nervous system. They were refined across hundreds of millions of years by organisms that had no brain but still needed to keep their cells running cleanly across long timescales. The same architecture that kept a bacterium viable is what keeps your neurons viable now. This is what it means for neurons to persist. Every moment they exist, they are managing the molecular pressures that eventually become disease in other brains, under different conditions or longer timescales. They are managing the same amyloid-beta that in someone else's brain began accumulating years before any symptom appeared. They are maintaining the same tau that in another set of neurons lost its grip on the microtubules and began to form tangles. The difference between a neuron that will last your lifetime and one that will not is a difference in balance, in timing, in the genetic variables that tip the scales earlier or later. The mechanism is universal. The outcome is not. What holds the balance is not luck. It is the continuous work of systems that were built by billions of years of evolution to solve exactly this problem. The chaperone network and the proteasome, the autophagy machinery and the microglial patrol, all of them operating in coordination, maintaining the conditions under which neurons can remain neurons rather than accumulating the damage that eventually ends them. The work is invisible, it is continuous. And tonight, while you sleep, it will continue without asking anything of you. And here is what might be the strangest thought to carry into sleep. The organ working to keep that balance tonight is the same organ that has been listening to this episode. It has been describing its own failure modes. It has been naming the proteins that, in some of its own cells, it is already managing imperfectly. Your brain has been trying to understand what goes wrong with brains, using the very machinery that the disease could eventually take. The cells holding your memories are still working. If you have found this episode valuable, I would be grateful if you would consider subscribing. It helps more than anything else.